The origin of life, from dead matter to living worlds, how chemistry became biology on a violent young Earth. Imagine standing on the surface of Earth 4 billion years ago. You would not recognize this place. The sky is a haze of carbon dioxide and nitrogen, tinted orange by methane and scarred with the constant flash of lightning. The oceans are not blue, but a murky greenish brown, superheated in places by volcanic eruptions that tear through the thin, unstable crust. Asteroids still pummel the surface with alarming regularity, each impact powerful enough to vaporize entire seas. There is no oxygen to breathe, no grass beneath your feet, no bird song, no life of any kind, at least not yet. And yet, within this hellscape, something extraordinary is about to happen. Somewhere in those violent, churning waters, perhaps in a shallow, sun-warmed pond, perhaps in the superheated darkness of a deep-sea volcanic vent, simple chemicals will begin to interact in ways that cross an invisible threshold. Atoms of carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus will link together into molecules of increasing complexity. Those molecules will begin to replicate. They will begin to change. And from this purely chemical process, something will emerge that has never existed before in the known universe: life. This is the story of abiogenesis, the origin of life from non-living matter. It is one of the deepest and most consequential questions in all of science. It is a story that stretches from the philosophical musings of ancient Greece to the robotic explorers currently rolling across the red deserts of Mars. And while we have not yet answered the question definitively, the progress humanity has made in the last century is nothing short of astonishing. The ancient question, where does life come from? The question of how life begins is as old as civilization itself. For most of recorded history, the answer seemed obvious. Life simply appeared. Leave a piece of meat out in the sun, and maggots will crawl from it. Let grain sit in a dark corner, and mice will emerge. This idea, spontaneous generation, was not a fringe belief. It was mainstream science for more than 2,000 years. The Greek philosopher Aristotle, writing around 384 BCE, articulated this view with characteristic confidence. He observed that certain animals seemed to spring from mud, decaying matter, or dew. Eels, he noted, appeared in rivers without any apparent parents. Flies materialized on rotting flesh. To Aristotle and the thinkers who followed him, the living world was porous. The boundary between life and non-life was easily crossed, and nature could generate organisms from raw material whenever conditions were right. It was an elegant idea. It was also wrong, or at least wrong in the way Aristotle imagined it. But dismantling it would take centuries of careful experimentation. The first serious challenge came in 1668, when the Italian physician Francesco Redi designed a beautifully simple experiment. He placed pieces of meat in several jars. Some he left open to the air, others he sealed with fine gauze. The result was decisive. Maggots appeared only on the meat that flies could reach. No flies, no maggots. The organisms were not materializing from the meat itself. They were being deposited there by living parents. Spontaneous generation, at least for visible creatures, was dealt a significant blow. But the debate was far from over. The invention of the microscope had revealed an invisible world teeming with microorganisms, bacteria, protozoa, fungi, and proponents of spontaneous generation simply shifted the argument. Perhaps large animals required parents, they conceded, but surely these tiny, simple creatures could arise spontaneously from broth or decaying matter. It would take another 2 centuries, and one of history's most brilliant experimentalists, to settle the matter for good. In 1861, the French chemist Louis Pasteur conducted his legendary experiments. He boiled nutrient broth in specially designed flasks with long curved necks that allowed air to enter but trapped dust and microbes in the bends. The broth remained sterile indefinitely. No life appeared. But the moment Pasteur broke the neck of the flask, allowing particles from the air to fall into the broth, microorganisms flourished within hours. Pasteur's conclusion was unambiguous and resounding. Under current conditions on Earth, life comes only from pre-existing life. He called this principle omne vivum ex vivo, or life from life. Spontaneous generation was dead. But Pasteur's triumph created a profound new puzzle. If life cannot arise spontaneously today, how did it begin in the first place? If every organism requires a parent, who was the first parent? The question had not been answered. It had been sharpened into something far more precise and far more difficult. The man who would forever change our understanding of life's diversity also had a quiet intuition about its origin. Charles Darwin, whose 1859 masterwork on the origin of species explained how life evolves, was more cautious about how it began. But in a private letter written in 1871 to his friend Joseph Hooker, he allowed himself to speculate. But if, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed ready to undergo still more complex changes. Darwin was imagining something radical, a natural chemical process by which the building blocks of life could assemble themselves from simple ingredients, given the right conditions. He was, in essence, sketching the first scientific hypothesis for abiogenesis. But neither the chemistry nor the understanding of cellular biology existed yet to test such an idea. It would remain a tantalizing suggestion for more than 50 years. Then, in the 1920s, two scientists working independently on opposite sides of the world took Darwin's intuition and built it into a rigorous scientific framework. In 1924, the Soviet biochemist Alexander Oparin published The Origin of Life, a groundbreaking book that proposed a detailed scenario for how life could have emerged through gradual chemical evolution. Oparin argued that Earth's early atmosphere was fundamentally different from today's. It was a reducing atmosphere, rich in methane, ammonia, hydrogen, and water vapor, but lacking free oxygen. In this chemical environment, Oparin suggested simple organic molecules could form spontaneously. Over millions of years, these molecules would accumulate in the oceans, creating a rich primordial soup of organic compounds. Gradually, these compounds would interact, forming ever more complex structures, colloids, droplets, and eventually something resembling primitive cells. 5 years later, in June 1929, the British Indian biologist J.B.S. Haldane independently arrived at a strikingly similar hypothesis. Haldane emphasized the role of ultraviolet radiation, which, in the absence of an ozone layer, would have bombarded Earth's surface with enormous intensity, as the energy source driving prebiotic chemical reactions in the early oceans. He envisioned the seas becoming a hot, dilute soup of organic molecules, from which the first self-replicating systems eventually emerged. The Oparin-Haldane hypothesis was elegant, testable, and deeply influential, but it remained theoretical. What was needed was an experiment, a way to recreate the conditions of early Earth in a laboratory, and see whether the chemistry of life could truly arise from simple ingredients. That experiment came in as 1953, and it changed everything. 2. Part 3, Sparks in a Flask. The Miller-Urey Experiment. The setting was the University of Chicago. A young graduate student named Stanley Miller, working under the guidance of Nobel laureate Harold Urey, designed an apparatus that was deceptively simple in concept, but revolutionary in implication. Miller constructed a closed system of glass flasks and tubes. In one flask, he placed water, which he heated to produce steam, simulating the early ocean. The steam rose into a larger flask containing a mixture of gases, methane, ammonia, and hydrogen. Urey's best estimate of Earth's primordial atmosphere. Into this gas mixture, Miller fired continuous electrical sparks, simulating lightning. The gases and any products they formed were then cooled, condensed, and cycled back through the system, mimicking the water cycle. Miller let the apparatus run for a week. When he analyzed the brown, murky liquid that had accumulated in the flask, the results were electrifying. The water contained amino acids, including glycine and alanine, two of the fundamental building blocks of proteins and, by extension, of all known life. The Miller-Urey experiment was a sensation. For the first time, scientists had demonstrated that the basic molecules of life could form abiotically, without any biological intervention, under conditions plausibly resembling those of early Earth. The primordial soup was no longer mere speculation. It was chemistry you could watch happen in real time. In the decades that followed, Miller and others expanded on this work. Reanalysis of Miller's original samples, conducted after his death in 2007, using modern analytical techniques, revealed that he had actually produced more than 20 different amino acids and a variety of other organic compounds, far more than he originally reported. Other researchers demonstrated that similar reactions could produce sugars, lipids, and nucleotide bases under various conditions. In the 1960s, biochemist Sidney Fox took the next logical step, showing that when amino acids were heated under dry conditions, as might occur on volcanic rocks near the shore, they spontaneously linked together into protein-like chains called proteinoids. Even more remarkably, when these proteinoids were placed in water, they self-assembled into tiny spherical structures called microspheres that bore a striking resemblance to primitive cells. These microspheres had defined boundaries, could absorb material from their environment, and could even divide, exhibiting rudimentary cell-like behavior without any genetic material at all. The picture was coming into focus. Simple chemistry, driven by natural energy sources, could produce the molecular building blocks of life. Those building blocks could spontaneously organize into more complex structures. But a crucial piece of the puzzle was still missing. How did mere chemistry become biology? How did molecules begin to carry information, to replicate, to evolve? The answer, many scientists came to believe, lay in a remarkable molecule, RNA. When molecules learn to copy themselves, every living cell on Earth today runs on a division of labor. HT stores genetic information. Proteins, HT including enzymes, do the chemical work of the cell, catalyzing reactions, building structures, transporting molecules. And beta RNA serves as the intermediary, carrying instructions from DNA to the protein-building machinery. This arrangement poses a classic chicken and egg problem. DNA needs proteins to replicate, but proteins need DNA to be made. Which came first? For decades, this paradox seemed intractable. The breakthrough came from an unexpected direction. In mice, 1967, the American biologist Carl Wosak, who would later revolutionize our understanding of life's family tree by using ribosomal RNA to classify organisms into 3 domains, proposed a radical idea. Perhaps our RNA came first. Perhaps in the earliest stages of life, RNA served as both the genetic material and the catalyst, performing both roles that DNA and proteins share today. At the time, the idea seemed almost fantastical. RNA was known as a passive messenger, a molecular go-between. The notion that it could act as an enzyme, a catalyst capable of speeding up chemical reactions, was pure speculation. Then, in C-1982, Thomas Cechik at the University of Colorado made a discovery that shook the foundations of molecular biology. Working with the single-celled organism, Cech found that a piece of RNA could splice itself. It could cut out a section of its own sequence and rejoin the remaining pieces, all without the help of any protein enzyme. He called these catalytic RNA molecules ribozymes ejects. 1 year later, in 1983, Sidney Altman at Yale University independently discovered another catalytic RNA, an RNA molecule that processes transfer RNA in bacteria. The implications were enormous. RNA was not merely a passive carrier of information, it was an active chemical agent, capable of catalyzing reactions just like protein enzymes. Cech and Altman shared the CEF 1989 Nobel Prize in Chemistry for this discovery. In CEF 1986, the Harvard molecular biologist Walter Gilbert gave this emerging paradigm a name, the Cech RNA world. Gilbert proposed that there was an ancient era in Earth's history, before DNA, before proteins, in which RNA molecules performed all the essential functions of life. They stored genetic information in their nucleotide sequences. They catalyzed the chemical reactions necessary for metabolism. And crucially, they could replicate themselves, providing the raw material for Darwinian evolution at the molecular level. The German biophysicist Manfred Eigen had already laid the theoretical groundwork for this idea. Eigen developed mathematical models of molecular self-organization, describing how populations of self-replicating molecules could form cooperative networks he called hypercycles. In these hypercycles, different RNA molecules would catalyze each other's replication, creating a self-sustaining system that could grow, compete, and evolve, all without cells, without DNA, without proteins. But the RNA world hypothesis faced a formidable chemical challenge. RNA nucleotides, the individual units that chain together to form RNA, are complex molecules, and scientists struggled for decades to show how they could form under prebiotic conditions. Many critics argued that RNA was simply too sophisticated to have been the first self-replicating molecule. Then, in 2009, British chemist John Sutherland achieved a landmark breakthrough. Working at the University of Manchester, Sutherland demonstrated a prebiotically plausible synthesis of activated pyrimidine ribonucleotides, a section to 2 of the 4 building blocks of RNA. His synthesis did not require preformed sugars or bases. Instead, the components assembled through a series of reactions, starting from simple molecules like cyanamide, glycolaldehyde, and phosphate, all of which could reasonably have existed on early Earth. The reactions were driven by ultraviolet light and proceeded in water, precisely the kind of conditions Haldane had imagined decades earlier. Sutherland's work did not prove the RNA world hypothesis, but it removed one of its most significant obstacles. Meanwhile, researchers like Jacques Jostak at Harvard were making parallel strides in creating protocells, simple, membrane-bound vesicles containing RNA that could grow, divide, and even undergo a primitive form of natural selection. By 2019, multiple research groups had created protocells capable of growth and division using simple chemical systems, advancing our understanding of the critical transition from chemistry to biology. While the RNA world captured much of the spotlight, a competing hypothesis was gaining ground, one that placed life's origin not in a sunlit pond, but in the crushing darkness of the deep ocean floor. 1977, a team of geologists led by Beestjack Corliss and John Edmund made a breathtaking discovery near the Galapagos Rift in the Pacific Ocean. Diving in the submersible Alvin Gam, they found hydrothermal vents Gam, fissures in the ocean floor where superheated, mineral-rich water poured into the frigid deep sea, and surrounding these vents, in total darkness, thousands of meters below the surface, they found thriving ecosystems, giant tube worms, clams, shrimp, and dense mats of bacteria, all sustained not by sunlight, but by chemosynthesis, the harvesting of chemical energy from hydrogen sulfide and other volcanic gases. The discovery was revolutionary. It demonstrated that life did not require the sun. It could be powered entirely by the chemical energy of the Earth itself, and it immediately suggested a new possible cradle for life's origin. In 1993, geochemist Michael Russell and his colleagues formalized this idea into the alkaline hydrothermal vent hypothesis. Russell proposed that life did not originate at the scorching, acidic black smoker vents found near mid-ocean ridges, but rather at gentler alkaline vents, structures where warm, hydrogen-rich alkaline fluid seeps into the mildly acidic early ocean. At the interface between these two chemistries, Russell argued, natural proton gradients would form across thin mineral walls, gradients strikingly similar to those used by all living cells today to generate energy through a process called chemiosmosis. In Russell's model, the porous mineral structures of alkaline vents acted as natural compartments, tiny chambers where organic molecules could concentrate and react. Iron-sulfur minerals within these walls could serve as catalysts, driving the reduction of carbon dioxide into simple organic molecules. Over time, these mineral-catalyzed reactions could give rise to increasingly complex chemistry, eventually producing the molecular machinery of life. Russell's hypothesis received a dramatic boost in the year 2000, when marine geologist Deborah Kelly and her team discovered the Kali Ost-City hydrothermal field on the mid-Atlantic ridge. Unlike the violent, short-lived black smokers, Lost City was an eerie forest of tall, white carbonate chimneys, some reaching 60 meters in height, fed by slow, warm, alkaline fluids rich in hydrogen and methane. The chemistry was remarkably close to what Russell had predicted, a real-world analog for his origin of life model, sitting on the ocean floor like a natural laboratory that had been running for tens of thousands of years. The hydrothermal vent hypothesis and the RNA world are not necessarily incompatible. Some researchers envision a scenario in which the basic organic molecules formed in vent environments, while RNA-based replication evolved later, perhaps in different settings. The truth may involve elements of both, and perhaps of scenarios not yet imagined. While chemists and biochemists work to recreate life's origin in the lab, geologists search for its fingerprints in ancient rock. The oldest widely accepted evidence of life on Earth comes from fossilized stromatolites, layered structures built by communities of microorganisms, found in rocks from Western Australia dating to approximately 3.5 billion years ago. But in 2006, Alan Nutman and his team reported an even more extraordinary find: stromatolite-like structures in 3.7 billion-year-old rocks from the Isua Greenstone Belt in Greenland. If confirmed, these structures would push the evidence for life back to a time when Earth was barely 800 million years old, still cooling from its fiery formation, still being bombarded by asteroids during the late heavy bombardment. The implication is staggering. Life may have arisen on Earth almost as soon as conditions allowed. This suggests either that the origin of life is a relatively easy process. Given the right chemistry and the right conditions, it happens quickly, or that life originated elsewhere and was delivered to Earth, a hypothesis known as panspermia. The panspermia debate received a dramatic injection of public attention in 1996 when a team led by David McCaig at NASA announced the discovery of possible microfossils in Pastor ALH 84001, a meteorite that had been blasted off the surface of Mars by an ancient impact and eventually fell to Earth in Antarctica. The structures were tiny, far smaller than any known terrestrial microbe, and the claim remains highly controversial. Most scientists today consider the evidence insufficient to confirm Martian life, but the announcement galvanized the field of astrobiology and underscored a profound point. The question of life's origin is not confined to Earth. We live in an extraordinary moment in the history of this ancient question. For the first time, humanity possesses the technological tools to search for life beyond our planet, and we are using them. On Mars, NASA's Perseverance rover, which landed in Jezero crater in February 2021, is methodically drilling into ancient lakebed sediments and sealing rock samples for eventual return to Earth. Jezero crater was chosen precisely because it preserves a river delta, a geological environment where, on Earth, evidence of microbial life is commonly found. Perseverance carries instruments specifically designed to detect biosignatures, organic molecules, mineral patterns, and chemical anomalies that might indicate past biology. Beyond Mars, attention is increasingly focused on the sexy moons of the outer solar system. Jupiter's moon, Europa, and Saturn's moon, Enceladus, both harbor global oceans of liquid water beneath their frozen surfaces, kept warm by tidal heating. In 2005, the Cassini spacecraft flew through plumes of water vapor erupting from Enceladus and detected organic molecules, molecular hydrogen, and silica nanoparticles, exactly the chemical signatures expected from hydrothermal activity on the moon's ocean floor. NASA's Europa Clipper mission, launched in 2024, will conduct detailed flybys of Europa to assess its habitability. And then there is the E. James Webb Space Telescope, launched in December 2021, which is now analyzing the atmospheres of exoplanets, worlds orbiting distant stars. By studying the light that filters through a planet's atmosphere during transit, JWST can detect the spectral fingerprints of specific molecules, water vapor, carbon dioxide, methane, and potentially age biosignature gases like oxygen or dimethyl sulfide that could indicate biological activity. We are not yet able to detect life directly on these worlds, but we are narrowing the search, identifying which planets merit closer scrutiny with future, even more powerful telescopes. Perhaps the most audacious line of origin of life research involves not finding life, but making it. Scientists in the field of synthetic biology are attempting to construct minimal living systems from scratch, the simplest possible collection of molecules that can self-replicate, metabolize, and evolve. This approach attacks the origin of life question from the opposite direction. Instead of working backward from modern organisms, trying to strip away billions of years of evolution to find the original core, synthetic biologists work forward from chemistry, trying to build up to the simplest system that qualifies as alive. Researchers like Ax Szostak have made remarkable progress in constructing protocells, vesicles made of simple fatty acids that can encapsulate RNA, grow by incorporating additional lipid molecules from their environment, and divide when subjected to mechanical stress. Other groups have engineered RNA molecules that can catalyze the copying of other RNA molecules, approaching the critical milestone of self-sustained RNA replication. The goal is not merely academic. If scientists can identify the minimal chemical requirements for life, they will have established the threshold between chemistry and biology, the precise point at which a collection of molecules stops being a mere chemical system and starts being a living organism. This knowledge would have profound implications for estimating how common life might be in the universe. If the chemical threshold is low, if life emerges readily from common ingredients under common conditions, then the cosmos may be teeming with biology. If the threshold is high, if the origin of life required an extraordinarily improbable sequence of events, then we may be far more alone than we imagine. Mas T. Part 9. What we know, what we don't, and why it matters. Let us take stock of where we stand. We know that the basic building blocks of life, amino acids, sugars, lipids, and nucleotide bases, can form spontaneously from simple chemicals under conditions that plausibly existed on early Earth. The Miller-Urey experiment and its many successors have established this beyond reasonable doubt. We know that these building blocks can self-assemble into more complex structures: proteinoid microspheres, lipid vesicles, short RNA chains. We know that RNA can serve as both a carrier of genetic information and a catalyst of chemical reactions, supporting the RNA world hypothesis as a plausible framework for the earliest stages of molecular evolution. We know that natural environments, from warm little ponds to alkaline hydrothermal vents, could have provided the energy, the raw materials, and the compartmentalization necessary for prebiotic chemistry to flourish. We also know that life appeared on Earth remarkably early, possibly within the first billion years of the planet's existence, suggesting that the transition from chemistry to biology, while not yet understood in detail, may not require as extraordinary a set of circumstances as once thought. What we do not yet know is the precise sequence of events that gave rise to the first living cell. We do not know whether life began in a warm surface pond, a hydrothermal vent, a volcanic hot spring, an ice-covered ocean, or some other environment entirely. We do not know whether RNA was truly the first self-replicating molecule, or whether some simpler, now extinct molecular system preceded it. We do not know whether life arose once and endured, or whether it arose multiple times. But only one lineage survived, and we do not know whether life has arisen elsewhere in the universe, though we are, for the first time in history, equipped to look. These are not gaps in knowledge to be frustrated by. They are frontiers to be explored. The origin of life sits at the intersection of chemistry, biology, geology, planetary science, and astronomy. It is one of the most profoundly interdisciplinary questions in all of science, and the pace of discovery is accelerating. Epilogue: the most important question. There is a deeper reason why the origin of life matters, beyond scientific curiosity, beyond the search for extraterrestrial biology, beyond the technological applications of synthetic life. The question of how life began is, at its core, a question about what we are. Are we the inevitable products of natural law, patterns that chemistry is bound to produce whenever conditions allow? Or are we cosmic accidents, the result of an improbable chain of events that may have occurred only once in the entire history of the universe? 4 billion years ago, on a young and violent planet, something crossed the line from non-living to living. From that first flicker of molecular self-replication, in a warm pond or a deep vent or some setting we have not yet imagined, arose everything, every bacterium, every tree, every whale, every human being, every thought and every question, including this one. We do not yet know how it happened, but we are closer than ever before. And the search itself, the act of asking how dead matter came alive, may be the most remarkable thing that dead matter turned living has ever done. The investigation continues in laboratories, in ocean depths, on the surface of Mars, and in the light of distant stars. The origin of life is not a question that belongs to the past. It is a question that is being answered piece by piece right now.